In this video, I'm going to walk you through post-lab 34 spectral synthesis and spectral analysis of aspirin. Question number 1 and 2 have to do with IR analysis. For question number 1, I want you to attach your IR and a reference IR for aspirin. Your reference IR can come from NIST or SDBS or any other site that is reputable. And on your IR spectrum, I want you to identify significant similarities between these two spectra. Obviously, the IR that you obtain and the reference IR, they're not going to be exactly the same because they come from two different machines, but there should be similarities between the two spectra that allow you to verify that your IR is in fact of the aspirin molecule. And then for question number 2, I want you to get a reference IR for salicylic acid from one of the same sites, and I want you to identify differences between your aspirin IR and the reference salicylic acid. So this is a way of verifying that you did in fact convert the salicylic acid into something else. Your IR and the reference for salicylic acid should be pretty different because they are of two different molecules. Question number 3, describe the results of the ferric chloride test. This is just describing what it visually looked like to you and what that meant. For question number 8 is asking from your textbook, question number 4, exercise 8, is asking you to compare aspirin versus salicylic acid in terms of which one is the stronger of the two. You have a chemical reaction on page 288 at the bottom of the page, and that chemical reaction is giving you the structure of salicylic acid. It's also showing it that, so the reaction is salicylic acid plus acetic anhydride, making aspirin and acetic acid. And in this balanced equation, it's giving you the structures of salicylic acid and aspirin, so you could use that to help you answer this question. You could also just use the chemical reaction to help you answer the question. Keep in mind that reactions always move away from our strong acid, I shouldn't abbreviate it, SA, strong acid and strong base. Strong means the same thing as reactive. So our strong acids and our strong bases are always going to be reactants on the left-hand side of our chemical equations. Now, your last question here is exercise 5 from the textbook. And this is going to be a problem that you're going to do multiple times throughout the year. The question itself is actually on the next page because it's big. You're going to create this flow diagram for the chemical reaction. All of these boxes need to be filled in with one thing from this list. Notice that a lot of the things are on the list more than once. That means that they're going to appear more than once in this diagram. So you're really going to want to refer to the procedure in the textbook to help you fill this out. But basically what you're looking at here is the chemical reaction that's written this way instead of from left to right. That's just to make it fit better on the page. So up here in this first spot, you're basically going to be writing in all of your reactants, all of your starting reagents, everything that you started with. Think about all of the different things that you put into that flask. And then this means that a reaction took place. So in this spot, you're going to be writing in all of your products and also any byproducts, any solvents that didn't get used up. They may not always show up. Solvents consumed. Solvents may not always be included in these diagrams, but any solvents that are not consumed are going to show up in here as well. When you're filling this out, it's helpful to look at the procedure. It's also helpful to refer to the chemical equation, the balanced chemical equation for the reaction, to help you identify any of the byproducts. And then from all of this big mix of products and byproducts, you did a vacuum filtration. And in the vacuum filtration process, that's a separation technique. So I'm going to draw poorly drawn Erlenmeyer flask there. And here's your little filter funnel inside the Erlenmeyer flask. And you poured this huge mixture of stuff. You poured that into the Erlenmeyer flask. And something collected up here as a solid. And everything else ended up down here in solution in the bottom. So this one thing that you isolated all by itself. This vacuum filtration is trying to show that you're separating this mixture of things into two spots. So this little guy is going to end up all by itself, and all of these things that made it through the filter funnel, they're going to end up over here. When you're doing vacuum filtration or really any type of separation technique, everything that is present prior to the vacuum filtration also needs to show up after the vacuum filtration. This is not a chemical reaction, so nothing is being converted during the filtration process, meaning that whatever you write in this box needs to show up in one of these boxes down here, and whatever you write in this box, same thing. You can't have any different substances from as you move from these 4 boxes down to these 4 boxes.